Welcome to the Chalk Talk Gym podcast, where we explore insights into healthcare that help uncover new opportunities for growth and success. I'm your host, Jim Jordan. Welcome to Chalk Talk Gym. In today's episode, we are honored to have Dr. Chris Apfel. He's a distinguished physician scientist whose personal experience with his parents' battle against cancer has driven him to revolutionize cancer treatment. He's the co-founder of Sage Medic Corporation, a pioneering company dedicated to developing precision medicine oncology solutions. His innovative approach involves using a unique microtumor model to predict the most effective cancer therapy to apply to the particular cancer, offering hope to patients with even the most challenging cases. In this episode, we delve into the intricacies of cancer treatment, the evolution of precision medicine, and how Dr. Apfel's groundbreaking work is transforming patient care. Doctor, tell me and the audience a little bit more about yourself and how this technology evolved.
It's my real pleasure to be with you on this podcast. I am Dr. Chris Apfel. I'm a physician scientist whose parents actually have been affected by cancer. And when my father experienced cancer, he refused therapy because when he took care of my mother who had ovarian cancer and the treatment didn't work in her last months of life and it made her only more miserable, he said to me, unless you can promise me that the treatment is going to work, I don't want it. And I was, wait a minute. I thought we are now in the era of precision medicine. I thought we know what we are doing. We spent billions of dollars in cancer treatment, and it was only then that I realized that cancer treatment, even today, is, in layman's terms spoken, a hit or miss. In a late stage 4 lung cancer, tumor response rates are as low as 30%. And then it's often only temporary, so it gives you only 2 or 3 more months on average. But you never know whether the treatment is going to work. And I said, how can that be? So as a physician who has also been active in the intensive care medicine space, we know that if a patient has an infection, we usually cultivate the germs and we then put on antibiotics to understand which therapy is likely to work best. And so I asked my oncologist, why are we not doing this for cancer? Can we not try it out on the tumor itself instead of trying it out on the patient? And the response I got was, people have tried it before, but hasn't worked out yet. And that didn't make sense to me. So I actually looked deeply into the literature, spent over a year actually to review what has been done in the field. And there have been honest efforts. But it was clear that there were some biological misconceptions. There were also some other strategic limitations. There was often that it took too long to grow the tumors. And so what we realize is somebody needs to step up to the plate and really develop a method where we can know what's the most effective treatment for a cancer patient. And I think we have now, with the support of my wife and I, we founded this together. She's originally also a pulmonologist. She's now also a psychiatrist as well. And when she was a pulmonologist, she actually has treated also a lot of lung cancer patients. So she has experienced also firsthand how difficult it is to find the most effective treatment. And that's what we set out to do.
So the company is called Sage Medic?
Yes.
And when did you start it?
The Sage Medic Corp was incorporated 2017. And we started this on our own dime. It was clear that we would be unlikely to get NIH funding. And so we needed to develop a technology that is robust, that is IP protected, and where we can actually know this can reliably predict the most effective therapy for cancer patients.
So how many cancers does your test cover?
So the advantage of the Sage Oncotest is that we can cover a very wide range of tumors because basically all solid tumors. And we actually have also done some multiple myeloma and some lymphoma patients. The advantage of our technology is that we take a fresh biopsy and we divide up the tumor into fragments, re-aggregate them within a day, And then we have hundreds of these microtumors that are similar to your original tumor and therefore can then test them within 3 to 5 days, understand which therapy is likely to be most effective, and read that information back to you and your oncologist. And so a wide range of tumors, all solid tumors. We're talking about ovarian, we're talking about glioblastoma. We also can take, and that's a great interest, tumors of unknown origin, rare diseases, rare cancers, because there aren't enough studies that guide which therapy might be effective. And so we can test a wide range of tumors, all solid tumors. It's basically our platform is tumor type agnostic.
Okay. So when you take the biopsy, one of the challenges out there is hitting the exact tumor. What's the margin of error on the selection of the sample size? How does that influence the ability for you to detect what cancer is there and what isn't there?
So there are two questions in here, right? So how much tissue do you need? And then the other question is, how accurate is the prediction, right? So how much tissue we need. If you are scheduled for surgery, let's say ovarian cancer or glioblastoma, there is usually a lot of tissue that otherwise would go into the bin, into the trash. And that's fresh, valuable tissue before it's thrown into formalin that actually needs to be prearranged to get to us. Then there is usually a ton of tissue available. Anything above a gram is enough to test dozens of drugs. If you already have been diagnosed, if you already had your standard of care treatment and the treatment doesn't work for you, then a fresh biopsy makes sense. That could be from a lymph node. The lymph node could be resected on the neck or elsewhere when it's easily accessible. It could also be a core needle biopsy. So we have received a liver biopsy from breast cancer patients. That's a possibility. Those core needle biopsies provide limited tissue, but because our technology is so advanced, we can test with those at least 6 to 12 drugs. And so either a core needle biopsy or a surgical biopsy is sufficient to test the most important drugs.
So you and I had nerded out about a month ago on a conversation, and I think I told you of this company I knew in the early days, and the data went something like this. If you treated the right chemotherapy to a right cancer, you got a certain result. If you used the wrong chemotherapy to that cancer and then used the right one secondarily, the results were very different. Can you explain that concept to the audience?
So we have actually looked into the literature, have done our own meta-analysis. In principle, what we do know is if you get the best possible treatment, you basically double your chance that your tumor will respond to the treatment. That's clear. To what degree it translates into surviving, that's probably not doubling the chance. It's probably increasing it by 40%. But 40% in the field of oncology is a big deal. And that's often not obtained by any new drugs that's in development. In terms of accuracy that you were asking ahead of time in the previous question, the advantage of our assay is we have an extreme drug resistance assay. We dose those microtumors in different concentrations. And if the microtumors are not responding to very high dosages in vitro, so in our lab, they are very unlikely, more than in the high 90s of percentage, very unlikely to respond to a patient in the patient. So if you cannot kill it in the lab, it will certainly not work, most certainly not work in a patient. And that's a very important information because more than 50, 60, 70% of drugs are actually not effective for that particular type of tumor. And you want to know that ahead of time. Just knowing that can already double your chance. And then we have a graded response where we are also looking at what are the most promising options that are looking most promising, and that will be played back to the oncologist.
I guess before we continue, we should probably get some definition. So one of the things in the diagnostic world is this concept of sensitivity and specificity. Can you explain that? Because not everyone in our audience is from the diagnostic world.
Yeah, so the sensitivity is how many patients that actually will respond to the treatment are correctly identified to respond to that treatment. And the specificity is how many patients who do not respond to the treatment will be correctly identified not responding to that treatment.
Okay. And so for your test compared to other attempts in the past to do this, what's the difference?
The huge advantage of the Sage Oncotest is, number one, it's a 3D microtumor model. So it has the tumor microenvironment and the tumor heterogeneity of the patient's biopsy, and therefore is really a step ahead of what has been done before in the past. And because we are culturing the tumor, so we don't need to grow it, we have a result within 7 to 10 days. And it's not only the speed, it's also the test results, the likelihood to get a test result. In the past, when people have tried to grow tumors, the so-called clonogenic assay or the tumor stem cell assay, very often, if you don't have a very aggressive tumor, those don't grow, and then you don't have a result. And even if they grow, it may sometimes take 3 weeks, 4 weeks, 6 weeks, 2 months, 3 months. And the advantage of the Sage Oncotest is that we almost always have a reliable result for you.
That's amazing. So this brings about the concept of fitting into the bigger healthcare concept of precision medicine, correct?
That's right. And it's really an important step forward and brings it to the next level. In the last 10 to 20 years, there have been enormous advances in terms of genomic testing, next generation sequencing to identify certain mutations that are relevant driver of the tumor behavior. And if a patient has that mutation, or if the tumor harbors that mutation, then Big Pharma have been able to develop targeted therapy that can really target that particular mutation and often keep the tumor at bay. That is really a great success in terms of, instead of 30%, response rates can be as high as 60, 70, or 80%. Not 100%, but much, much higher than unspecifically with chemotherapy. However, so far only 1 out of 4 patients have mutations that are actionable, which means in 3/4 of the patients, roughly 75%, there are no actionable mutations. And then there is no information as to which therapy is most likely to work best. And that's where Sage Medic comes in, because we are independent of any mutations, of any potential pathways. We actually see what works best in a more holistic sense, because we take the tumor as a live tumor, because the sum is much more than, the whole is much more than the sum of its parts. And therefore, we can actually see whether a drug is working irrespective of which pathway might be affected. Being able to provide information for the majority of patients where genomic testing does not provide information, that's a big deal for many patients.
It's actually quite interesting. So maybe trying to figure out an example here for the audience, but I think of PDL1, I think of BRCA1 and 2. I think of different ways that people have said, if you have this, then you can use this product. But in your case, you could ironically discover that something that might be used for one of those areas actually works on a particular tumor. And without this technology, we might have dismissed the ability to use that drug.
Well, that's right. So there is actually a potential for discovering that certain targeted therapies that would normally not be considered for your tumor type, because there haven't been studies done on this tumor type, that they actually may be beneficial for you. And the advantage is that we are not, because we are looking in a holistic sense, in a very integrative sense on the totality of the tumor and not at a single mutation, there may actually be a tumor type that is actually does not harbor the mutation but would still respond to that targeted therapy.
That is actually quite profound. Is the pharmaceutical industry discovered this with you to try to quantify this or understand it? Or is it still early in the cycle of development?
We have been approached by a couple of companies. We are now working with 2 companies specifically that have a drug in development. It's currently pre-phase 1, so it's too early for a patient to have access to that, but it can also help drug developers for their label extension and identify, there may be a specific other type of tumor that are more likely to be responsive that we didn't have on our radar before.
So that's a little deviation here for our startup people. You can create this test and you can do what you're doing. But if you step back and you think about it ahead of time and how you collect data and how you present data, you could actually have a data set and samples that would create another business model for the pharmaceutical industry for future targets.
It could be useful for future targets in drug development in lead compound generation. It could be used in phase 1 to demonstrate the correlation with the patient outcomes. And then it could be used to de-risk the phase 2 and phase 3 studies. And so that you can have a statistically significant result with a lower number of patients in a shorter period of time when you can use this as a companion diagnostic. But that's in the future. Mm-hmm. What we are very focused on is how can we help a patient today? If a patient gets diagnosed with a serious disease, often standard of care will only provide average results. I mentioned ovarian, I mentioned glioblastoma, a brain cancer. Those patients would be best advised to contact us well ahead of time of the surgery, because otherwise the tissue goes into the formalin and then it's dead and can't be used, because ours is a real functional profiling test. And then those patients where the therapy is failing, when first-line therapy may have worked perhaps initially, but then the patient's tumor has developed resistance, those patients shouldn't wait too long contacting us to see whether we can get a biopsy and a sample from them to understand which one is the most effective treatment.
So in a treatment world now that's moving from monotherapies to combination therapies, how do you dial that in your approach?
First of all, what's really important is to try to stay within standard of care, because even within standard of care, there are a couple of options. And so the Sage Oncotest can help the oncologist to provide, as I like to say, superior care within standard of care. This can be completely NCCN guideline compliant. And if you look at the guidelines, very often combination therapies are also part of the standard regimen. And we can look into what do the individual drugs do and what do the combinations do. If we think about colorectal cancer, if we think about pancreatic cancer, if we think about FOLFOX or FOLFIRINOX, these are combination treatments that have 5-FU, they have oxaliplatin, they have irinotecan, and we can look into what are the individual components that contribute to the tumor responding to the therapy.
It seems to me that if I were a patient and I was trying to get information on this whole approach, where would I go to find that? Do you have a website that's consumer-oriented and one that's physician-oriented?
So exactly. So we have a website, sagemedic.com, sage like the plant or the sage person you're talking to. Medic, not medical. So one word, sagemedic.com. And you can search there. There's information for patients and there is information for physicians. And you can then, on the patient's page, you can register and request more information about the Sage Oncotest. You can then also, once you have your information, you can also speak with your oncologist. The important thing is that you educate yourself first. You need to understand what's the prognosis, what are the options that I have. If you, for example, search the web, there is this Target Cancer podcast where I have presented with Dr. Chunecha, and that gives you a good background on what Sage Medic is doing and why a number of oncologists are very excited about what we do. But the most likely is, because this is very new, most oncologists have not heard of us, and you need to actually also be able to provide the oncologist with some of information. Perhaps the website is one part, but the part that's important for oncologists is to say this can be within standard of care, and it can be a additional tool in their armamentarium to identify the most effective treatment that goes beyond genomic testing. and may provide additional options we might not have considered.
So how much is this covered by insurance today if I were to do this?
We are now fully accredited by the Centers for Medicare and Medicaid Services to commercialize the assay, to provide this information for patients and oncologists, but it's currently not covered by insurance. Insurance coverage often lags well behind the innovation. And so you would actually need to speak with us what this would cost. It depends on what type of test your oncologist would like to have. There's a standard panel that's NCCN guideline compliant. NCCN stands for the National Comprehensive Cancer Network. So those are the main universities in this country, the main cancer centers. And we can stay within that framework. And then some patients actually opt for having, and physicians order additional testing. So for example, lovastatin or disulfiram or other drugs that are FDA approved for other indications have actually shown effectiveness against cancer, and they can be used in combination with it. If those are to be tested in addition, that would of course be a different pricing than if you are just limiting yourself to the standard Sage Oncotest.
Now, in the medical world, the doctor always has the right to use their judgment on these things, and if something is off-label or not. But it would imagine to me that I would be even comforted more to have evidence in addition to my judgment that it works. So I could imagine more and more people would love to have this in their arsenal.
And you're touching on a point of evidence, and the question is how much evidence is needed. As we are launching and are new to the field, we are starting with cohort studies and then subsequently with randomized controlled trials. But what we are also doing is we are giving patients a discount if they are willing to participate in our, I would like to say, registry trial. The patients get a 25% discount if they are okay with us using this information for our own R&D and quality control purposes. And that way the patients have a cost reduction and we collect data that demonstrates the correlation with the patient outcome.
What a nice way to do that. You've been in business with FDA approval for how many years now?
So we just launched effectively this year. Okay. And to be clear, this is a laboratory developed test. So it's not FDA approved. And the regulatory pathway here is the so-called CLIA accreditation. And CLIA stands for Clinical Laboratory Improvement Act. And this act was put into law in the 1980s, in order to make sure that if you get your blood tested, if you get your hemoglobin or your electrolytes tested in any lab, that these values are reliable. Mm-hmm. And so what we fulfill all those criteria. We fall under that regulatory category, and we have hundreds of pages on SOPs to be 100% compliant. So we are a California registered, CLIA accredited lab. So that's the regulatory approval that we have in order to be able to provide this information for patients.
What challenges are your organization facing now as you look to expand this? You've moved from the science now to getting into the scaling and the business. What sort of activities do you have going on and challenges that you have going on as the healthcare system is changing? For example, I believe this summer there's been some changes from CLIA standards at the FDA, and the FDA was trying to implement something early this summer, but I believe there's a lot of lawsuits going on. So you have regulatory things changing on you. But just what sort of give the audience a sense of what it takes to commercialize this?
Yeah. So the regulatory issue is we are not troubled by that. Individual labs are exempt, academic labs are exempt. So I'm not concerned about that. I think we have that well under control. And the other part is if there is a requirement for FDA approval, which usually requires outcome studies, which are important not only for regulators, but also important for us. We want outcome studies. So at the end of the day, whatever is needed for an FDA approval, if we ever need that, we will generate this evidence anyway. Mm-hmm. And so from a regulatory perspective, we are not concerned at all. In terms of market penetration, most oncologists, especially in larger centers, feel very much locked into what is standard of care. And that has to do with what's described in the NCCN guidelines. And most oncologists are trying to stay within those walls to be covered and to have, so that they're not open up for liability is one part. But the other part is actually reimbursement. Even if they stay within the NCCN guidelines, there's sometimes pushback from insurers because healthcare costs have actually spiraled out of control. And therefore, oncologists are always reluctant in going the extra mile. Many oncologists are reluctant in going the extra mile if it's not covered by insurance. And so if you want to have superior care, you actually need to have an oncologist who is on your side and is willing to go the extra mile.
So in one of the first clear companies that I worked with, we raced up to $5 million in revenue at no problem. And then we just paused. And there were two issues that caused that pause. One of it is not having enough data to get the reimbursement studies in. But the second issue was we did not realize that the sample process in the typical hospital has one pathway or one flow, and we needed it to take a different flow. And so for our audience, when you're implementing something new like this, it ended up being that we switched to a FedEx box because whatever the company was using had one process flow, and we were losing samples because they didn't follow the normal supply chain, quote unquote. And so in addition to going through all that science, ends up being little details like that have to certainly be attended to.
And we address this by providing a shipment kit, briefs this to the patient. And this is actually a box that actually keeps it cool for up to 72 hours. Perfect. Which brings actually up another interesting point. Because it's live tissue, it needs to be processed right away and it needs to be fresh. It's usually shipped overnight. But we have also Saturday delivery acceptance. So we process on Saturday. But there are situations where FedEx doesn't deliver it and you can only pick it up on Monday. Interestingly, our assay is so robust that, and we have shown this repeatedly, that even if there is a 3-day delay, we still get basically the identical results. And that's another thing where, from a logistics perspective, we are very proud of that our assay will always come up with a useful result.
And I brought that up just for that perfect story because it tells the audience you can get the science right, and then there's all these little operational details to get the business model to work and scale to support that patient. And so that's fantastic. So what's the biggest lesson you've learned on your journey so far with this?
The biggest lesson is if you want to make a difference in people's lives, you need to step up to the plate and you have to do it. You have to overcome all those little hurdles, but it can be done. And I think what I learned is that if you want to move the world, you need to be bold and you need to be systematic. You need to be rational. You need to listen to people and you need to have the patient's needs. You have to put that first. Put first things first, like in the book of 7 Habits of Highly Effective People. And if you follow your true north in helping patients, you will find a way to make that happen.
I imagine there's a mental aspect for the patient too, that having the benefit of knowing that something's been tested and had a result, I have to imagine that has some sort of psychological effect. Have you any data on that other than probably the experiences you've had with your customers so far?
We have not systematically explored that, and there are different type of people. There are people who would prefer not to rock the boat. And in a way, if you think about let's say there is a small melanoma, stage 1 melanoma, that needs to be cut out in total. It's way too small for us to look at it. The pathologist has to have the whole specimen to demonstrate that there's a free margin. Your cure rate is in the high 90s, right? So it's not necessary. The same applies for many very early, let's say DCIS, ductal carcinoma in situ of breast cancer patients. There's a little lump, can be removed surgically, needs to be removed in total. There is no need for the Sage Onco test. And then there is the other side. When you have an aggressive tumor, you want this to have a peace of mind. And so there are patients who really are comfortable, if they have an early stage disease, to leave it to the standard of care, because standard of care provides good outcomes. But there are patients, they know that the outcome is likely not very favorable, and if they want to have a real fighting chance to overcome this, then they take action into their own hand. And for them, it's very reassuring to have additional information, what can be done. And then there are these other situations where the side effects of the therapy is extreme. It doesn't really think the tumor is responding. And then there's a big question, should we continue with the treatment, especially when you have failed first and second line treatment? And here comes another part, and that is the more often you are getting treated and the tumor doesn't respond, the lower becomes the probability that it will respond to the next treatment. And the next treatment would usually also be with side effects. So the question on going on to treat or not to treat is important. And that's where the Sage Onco test comes in as an extreme drug resistance assay. So we can actually rule out ahead of time and say, no, these other treatment options that we might consider, they actually show multi-drug resistance. So don't go for it. And it's actually useful that a patient knows the truth. Some patients want to know it. They want to know ahead of time, is this going to work? And there are other patients who want to see that everything else is tried, and that's fine. But then we can actually rule out those that don't look effective and just focus on those that might have a fighting chance.
But I think to the point, the example you just gave, that a lot of patients that have that attitude don't realize that being treated with the wrong one first could reduce their overall probability.
Right.
The importance, I think, of what you're doing.
Right. It's another important point, approaching us ahead of time and not to wait until it's too late.
So as you look into the future of your, then in business 10 years from now, what have you brought or changed to the treatment of cancer?
What we see it in years to come is that this will evolve, become part of the new standard, will bring precision medicine to the next level so that we have a higher rate of cure. Currently, when a patient, let's say, comes with a late stage cancer, the chance to real cure may still be fairly low, and the difference we can make in terms of long-term cure may be limited. But the patient may actually, by getting the right treatment, have another year. And that's a big deal already. Now, as we become more established and we will have publications that demonstrate the benefit, oncologists would be more comfortable to use it at an earlier stage. And when used as an earlier stage, when you have minimal residual disease, we expect that we can also increase the cure rate so that, let's say, a mother with a stage 3 breast cancer can actually be there for her family and effectively survive and have a good life.
That's great. Is there anything else you'd like to share with the audience?
I appreciate the conversation. And I think the important part is spread the word. If you are or one of your loved ones is affected, check out sagemedic.com. I do think that Sage is very blessed also with having recently assembled a very strong scientific advisory board. For example, Dr. William Kans has joined our board as the chair of our scientific and medical advisory board. He was the past president of the Society of Surgical Oncologists, and he has also been the chief medical and chief scientific officer of the American Cancer Society. And so slowly but surely, the establishment realizes that we are addressing a really important need and that this is not a incremental benefit, but this can be really transformational for the field in years to come.
In addition, for the patient, bringing down the overall cost of cancer care, those last treatments, those last years of treatment become very expensive.
And that's right. And I think that's also where the Sage Onco test can help to have more effective and rational decisions, sometimes a inexpensive generic therapy could be equally effective or better than a $200,000 drug. And that's all what SAGE is about, to make sure patients get the most effective treatment and the most cost-effective treatment.
I'm going to maybe talk out of order of our podcast here as we're wrapping up. But to me, that would be a great peace of mind, because sometimes when you are offered something that's more generic, you sometimes think this is a pharmacy benefit play and there's the narrowing in cost savings. And it would be incredibly comforting to me personally to know that I have evidence supporting that selection. I would bring such a peace of mind.
Yeah.
Yeah.
I agree.
Thank you. Thank you so much for sharing this. I'm going to actually not only have your Sage Medic website in the notes, but for the audience, I'll also put the Target Cancer podcast in there, because I thought that was beautifully done.
Thank you very much.
Okay.
I appreciate our conversation, and I'm looking forward to connecting with you also in the future on other levels.
Thank you.
Thanks for tuning in to the Chalk Talk Gym podcast. For resources, show notes, and ways to get in touch, visit us at chalktalkgym.com.